Bueno, pues buenos días o tardes a todos y a todas. Muchas gracias a los que ya están llegando a nuestra presentación hoy del Seminario de Ciencias Biológicas que tenemos por parte del doctor Rob Lavigne que ahorita voy a presentarles. Hoy la presentación la vamos a tener en inglés, nos la va a hacer Rob. So I'm going to switch into English to present. So Rob, thank you for attending our invitation to the department's seminar. It is a great pleasure to have you here. And I want to introduce Rob. Rob, in fact, I want to mention that Marta Vives, one of our colleagues, cannot be here. She wanted to be here to present Rob and has had some communication with him before and told me to, I told you already, Rob, but I wanted to say it, to thank you for the times that you've interacted with her and with students and hopefully many new collaborations or other collaborations can emerge in the future. But today we have a very interesting topic that we haven't had anything similar during this semester seminar. And Dr. Rob Lavigne is going to talk about human phage therapy, taking it step by step. And Dr. Lavigne is currently a full professor in the Faculty of Bioscience Engineering in the University of Leuven and the head of Animal and Human Health Engineering group. Also, Marta stressed a lot when I asked her a little bit about Rob's work, that Rob, and please correct me, Rob, if I'm wrong, but that you have been involved in many projects on genomics of bacterial viruses, that in fact you were very involved in the International Committee of the Taxonomy of Viruses, which is, I guess, which makes you an expert on these topics. And we're very happy to have you here. Rob has, I don't a very long list of publications, so some of them of very high impact. I was looking at the courses you teach, and it's really interesting on biotechnology, bioinformatics, on gene technology. Sounds very, very nice. So we're very happy to have you here. And okay, the audience is yours, and thanks, Rob, for being today with us in the seminar.
No, thank you so much for the kind invitation, Martha, Andres, all of you. So indeed, my name is Rob Lavigne. Our lab is located in Leuven, Belgium, where we perform fundamental molecular microbiological research, inspiring new biotechnology and antimicrobial technologies. And today I'm going to talk about traditional phage therapy in humans, and in 4 steps I'm going to take you on how this is implemented in Belgium and how we contribute from a molecular microbiology side to this research. So the main question I get when I talk to a broader audience is, does phage therapy actually work? And I hope that's a question that I'll provide a nuanced answer to. But for us in Leuven, it's actually a question that has been asked for 100 years. We had Bruinhoog in 1921 writing an essay on the potential of phages infecting Staphylococcus, the therapeutic potential and the favorable results that he obtained 100 years ago. So today we're approaching this with the new molecular tools that are available to us and trying to make this actually work as a therapeutic alternative to MDs. So the first step I want to walk you through is the basic microbiology. As many of you know, a bacteriophage or a strictly lytic bacteriophage recognizes very specifically the components on the cell wall. And for example, Pseudomonas aeruginosa, which is a dangerous pathogen, phages recognize the type 4 pili, the LPS layer on the surface. And this is a first and very specific interaction that is occurring. The phage irreversibly attaches, injects its DNA, initial genes are being expressed, and this takes over the bacterial metabolism so that new phage particles are being produced and finally released. So from an application point of view, that means that if one phage is theoretically able to reach a site of infection, it will prey on the bacteria that are present, and we have a self-amplifying antimicrobial agent, which is a quite unique characteristic combined with that specificity. At the same time, there is a co-evolutionary arms race going on in which phages adapt to the bacterial cell, but in turn, the bacteria can have maybe mutations on the cell wall so that the phage no longer can attach, or has restriction modification systems, or the CRISPR-Cas immunity system, and many others to defend itself against phage predation. But then in turn, phages will develop new molecules to overcome these blocking mechanisms. So that's what I mean with this co-evolutionary arms race. And that's a key essential topic, that a phage in that sense is not static, but the bacteria can succeed in blocking the phage at any of these stages of infection. If we look historically to phages infecting Pseudomonas aeruginosa as a major multi-drug resistant pathogen, phages have actually been known for quite some time. They were introduced in the 1960s, the so-called Lindbergh set, that was expanded to 18 phages. And with the introduction of electron microscopy, it was observed that there was an enormous morphology diversity that was present. You had 8 different morphologies, 8 type clades of phages that were able to infect isolates of this bacterial genome, of this bacterial species, Pseudomonas aeruginosa. But actually, you have to realize that in 2005, only 4 genomes were available at that time. Yeah, when I started my research, this was still being done by Sanger sequencing. Now, most of the time, it's Illumina sequencing or even nanopore sequencing that's emerging, allowing us much more quickly to determine the entire genome of the phages, but also of the bacteria, of course. So when we look at the Pseudomonas phages, we were able, when we were isolating our own, we could indeed confirm that all these different morphologies were present, and this is all drawn to scale. So you have these giant viruses containing genomes of up to 280 kb, other phages having genomes of about 48 to 50 kb in size. And originally, they were classified based on their morphology. Cryo-EM reconstructions allow us to get a more detailed view on how they are looking. But now, primarily, they have been classified based on their genomic traits. So, and the immense diversity of phages that's present, all infecting these Pseudomonas aeruginosa strains, is also reflected in the genomes. Here you see one genome, and each arrow represents an individual gene. And some of these genes are structural. They translate to proteins that are a part of the virion particle. Marked in gray are genes with predicted functions. These are usually DNA polymerase genes and so forth. But still, the majority of genes in most phages that we have are unknown. They call this the viral dark matter. They have no discernible function, but since they are so densely coded, all of these genes serve an essential purpose. What's interesting, when we compare these different clades of phages, they have no homology whatsoever. No DNA homology that is present between the phages, no homology often even between proteins. So from a molecular point of view, these are completely distinct viruses that infect the same bacterial species. What's important to understand, and when assessing the in vitro efficacy of phages, we have to know how well they are killing Pseudomonas aeruginosa isolates. And this is a very detailed graph. But what I want to show you is that when we combine this phage, LUS19, this phage, LUS24, and this phage, LBL3, we are only able to kill 72% of isolates, all belonging to Pseudomonas aeruginosa. So that's a key limiting factor. The host range of bacteriophages is incredibly narrow. It's very specific to that extent that only you need a combination of phages to target the majority of isolates that you will find in a clinical setting. This is an incredible limitation that has to be taken into account, because it means that, unlike antibiotics, we need a diagnostic identification of the specific bacterial strain that is there required for treatments, and whether we have phages that are able to infect that specific isolate. Yeah. That often results in the combination of phages, which are simply brought together based on their host range to cover as much as possible the bacterial isolates within a certain bacterial species. But in general, when combining phages, there are no rational design rules that have been established that tell you which phages should be combined in a cocktail to more optimally address the majority of strains for a certain pathogen that are around. So this also limits the applicability to more chronic infections and makes it less amenable to urgent acute infections. So we're trying to take some steps towards rationally designed cocktails. And on one hand, you can test a certain phage represented here in a phage dilution series spotted on a lawn of a given bacterial isolate. And you see here that this PB1 phage is able to infect the bacterial cell because it results in a negative colony. Individual phage particles will infect and destroy these bacteria, resulting in these plaques or negative colonies, giving productive infections. So that's a very basic characteristic, but a very important one, allowing you to see, do they infect, yes or no? And this can be actually summarized into binary response variables, allowing you to look at individual phages and for an entire range of Pseudomonas aeruginosa isolates to see whether they infect, yes or no. Yeah. And then we actually are currently moving to something unexpected, namely marketing-based association rules to make sense of this. Yeah. You have been exposed to this, these marketing-based association rules, because stores will actually track for individual customers the items that they are buying and trying to come up with, based on the transactions that are occurring, whether someone is buying wine, cheese, ham, or bread, yes or no, trying to come up with rules of which things are frequently purchased together. Is there any exclusion factors between objects? And to be able to explore this data and visualizing it is key in this technique. So based on this analysis, for example, you can extract some rules that if someone buys wine, then they're likely to buy cheese. Yeah, if they buy bread, they're also likely to buy cheese, but bread and wine are not specifically associated together. So we can actually transpose this statistical methods to mine our results of the infectivity data to uncover specific association rules. For example, we can query, do phage 1 and 2 often infect the same host? Are some phages complementary in their infection profiles? To then extract some association rules and come up with which combinations based on these traits are best to combine. Yeah, this can be simply inferred that related PB1-like viruses often infect the same host. Often this VKMV virus infects strains that this PB1 virus cannot. So these things that are often just simply looked at and gazed on are now more formally brought together through this association rules, which will hopefully be a tool that we are putting out in the near future. More advanced approaches introduce machine learning to understand the phage-bacteria interaction, where we start from trained labeled examples having a high quality input to optimize certain learning algorithms, receiving a set of inputs along with corresponding correct outputs to then have an algorithm learn by comparing the actual output with correct output to find errors, and then in a final step to modify those models. Yeah. So what does that mean? Can we use this artificial intelligence to rank effective phages to see what determines a successful phage infection of a certain bacterial strain that has been sequenced? For this, we need more than just the genome information from the phage. We also need the genomes of the bacterial strains. Yeah. And we have, together with some collaborators, sequenced and obtained over 600 Pseudomonas aeruginosa isolates, which have been fully sequenced. And one of the basic traits that you can discern from them is when we take these, for example, 600 strains, how are they clustering together? In other words, what is the diversity of Pseudomonas aeruginosa on its own? Well, this diversity is incredibly large. We see 5 different clades of Pseudomonas aeruginosa strains that are present. And a lot of the genetic diversity of Pseudomonas aeruginosa makes the interactions with phages difficult to predict. When you take a subset here of strains, we can make a distinction between the core genome, meaning all of the genes that are present in all of the isolates there, versus the accessory genome, which contains genes that are specific to one or a subset of strains. And for Pseudomonas aeruginosa, this accessory genome is incredibly large, and this is what muddles and makes the interaction between phages and bacteria very unclear, or at least very unpredictable. What is incredibly important in the setup that we're trying to achieve is that we are, in fact, combining sequencing technologies to ensure that we have a fully closed genome. We're combining the short read sequencing of Illumina with long reads of nanopore sequencing, and then using hybrid assemblies to fully resolve, on one hand, the entire chromosome, and on the other hand, to identify plasmids that are present. And this is turning out to be really valuable to do this. Having the information on the mobile elements, the plasmids, the presence of transposons, the structural variation that can occur within the bacterial chromosome is really valuable because it also improves our capacity to predict or improve gene annotations. For example, prophages, which are non-lytic phages that lie dormant within the bacterial chromosomes. We can better predict those if we have hybrid assemblies, fully resolved genomes, compared to bacterial genomes that have only draft genomes made by Illumina sequencing. And when we delve deeper in this, we see that the genetic determinants for phage infectivity are often located on the accessory genome. We can, in our annotation of our bacteria, see is there anti-CRISPR proteins present, which CRISPR-Cas proteins are present, other phage immunity systems that have been recently discovered that prevent phage infection, and whether their presence is there or not. Having this as a good annotation is really important. And we see that many of these anti-phage systems are highly specific to certain isolates and therefore present in the accessory genome. So we have a double input now. We have on one hand the genomes of our phages. On the other hand, we have the correctly annotated genomes of our bacterial isolates, and we have the infectivity data. Yeah? So we're creating this custom database where we focus on the prophages that are present, the defense systems that are present, whether or not our phage is infecting. There is the functional annotation of all the genes that is present. And then we can link the phylogeny to specific traits that are present. The presence of prophages, CRISPR restriction modification systems, and tabulate this also on the host range, as I mentioned before, whether a phage is infecting, yes or no. And then we can start data mining, whether there is co-occurrence, feature selection, phylogenetic signals, and so forth. And the way we do that is by adopting a supervised learning approach that allows to model complex systems using a training data set. So what does that mean? We have for a certain trait the specific input properties. There is the biological process, which is, can a phage infect, yes or no? Yeah. We do that, and that generates an output based on a specific model. And for this, we tested over 270 models, including support vector machines, a regression random forest model. And actually, we have our infectivity data. So as input, we actually use 2/3 of all of the data, infectivity data that we have, to train our model based on these existing machine learning models to see how good is the output. Yeah? And then we use the remaining 1/3 as a test set to see how well the model has been trained, how well it works. Yeah? This includes both black box models, which are very flexible, but you get very limited information from them, to also white box models that allow better parameter interpretation. I realize this is a bit more technical for the biologists among you, but allow me to illustrate it with just one relatively well model. It's a white box model with an accuracy of 80%. And what does that mean? That when we look to the actual data, yeah, there are 4 quadrants that are happening. Either a strain is infected or is not infected. And this is based on the biological data. We see that for a certain phage, most strains are not infected. Some strains are infected. And when we apply our machine learning model, how well does it match? Yeah, the machine learning model says, okay, these strains are infecting, these are not infecting. and so forth. So in this case, having the 660 not infecting and the 195 infecting, those are correct interpretations by the machine learning model. And these values are not super high, but they already give us some indication. And the interesting part about this model is that we can actually see what is determining to the correct, what is contributing, what is most important for the model to say, okay, this strain will infect, this strain will not infect. And indeed, we can look at which phage proteins are important for infection, which host protein, the presence of which host proteins are important. And this allowed us to see the correlation between infectivity and CRISPR-Cas. It allows us, obviously, to identify structural phage proteins that impact the infection process, which is quite logical. It also allowed us to identify potentially new phage receptors that are being used by some phages to infect Pseudomonas aeruginosa. These models are promising. They are not perfect yet, and we're working to improve on them in the future. But I think in general, and in this first part, I hope it's clear that we have an immense microbial diversity of both the phages and the Pseudomonas cells, which represent a massive challenge in understanding the highly specific nature of the interactions that exist between the phage predator and the prey bacteria. Understanding this molecular background in a profound way, I think, is key in understanding and therefore implementing phage therapy in a successful way. Thirdly, these new statistical and bioinformatics tools, including machine learning, can help us make sense of huge amounts of data. Either you're confused by not having enough data or you're confused by having too much data. It's only in this middle ground we usually work, and machine learning can help us to make sense of these vast data sets, but it always relies on accurate, reliable, well-organized data, meaning in this case, well-sequenced bacterial genomes, well-annotated, and having reliable microbiological data to support it. In a second step, and this is going much more towards the biology side, we're looking at the preclinical assessment of bacteriophages, either in vitro, in vivo, and towards processing. So one assay that we described and set up is actually querying what is the in vitro efficacy of phages, not on an agar plate, but in a more relevant environment like this airway surface liquid model. So instead of using a basic agar plate, in this model, there is actually airway epithelia, either from a CF patient's epithelial cells or normal epithelial cells, but the CF epithelial cells create this hyperviscous mucus layer. And this we can insert in this mini cell hanging cell cultures and apply Pseudomonas aeruginosa to it, see how they actually infect this in vitro epithelial cells, and see how phages can protect this or reduce at least the bacterial numbers of Pseudomonas aeruginosa that's present. So this we did not only for normal human epithelial cells. My computer, yes. And we did this for two phages. On one hand, this phage called KTN4 and this giant phage PA5-OCT. And the controls, you see that the bacterial count of 10 to the 10th is actually radically reduced by the treatment of phage by several log units in this in vitro assay. For KTM4, this was highly efficient, for this PA5-OCT, that activity was drastically lower, I would say. So the phages themselves, different phages, are going to have a different effect on this model. Yeah. In addition, when looking at CF, patient epithelial cells, yeah, here we saw that the effect was also more pronounced. And another effect that I want to draw your attention on is that besides the two phages that we used, we actually tested this on 3 different Pseudomonas aeruginosa isolates. A lab strain, PA01, a CF strain, 708, and a normal strain. And there you also see that not only the phages is a factor, but also the specific strain can be more efficiently eradicated compared to other bacterial strains, all making it incredibly specific. Yeah. So the phages can protect these epithelial cells lines of normal and CF origin, but the efficacy is strain dependent and phage dependent. This is just as a brief introduction, the in vivo efficacy of Pseudomonas phages in an animal model. This is not research by our group, but by the group of Laurent Debarbieu at the Institut Pasteur, where they engineered Pseudomonas aeruginosa PAC strain to become bioluminescent. And this bioluminescent strain was placed on the nose of the animals here. And when you put them under a special microscope, you can see the luminescence as a function of time. And you can see the more red, the more bacteria that are present. And comparing phage treated versus non-treated samples. After 2 hours, you can see that in both groups, the nose is colonized. After 4 hours, you can see that from the nose, the bacteria have begun migrating to the lungs. And here these lungs are already very well infected. But you see here that the treatment of phages after 2 hours is already clearing the nose that is now at least visibly free of bacteria. At 6 hours, Debarbieu and his colleagues noted that in the control group, both lungs were infected, whereas in the treatment group, phages were eradicating the bacteria. After 48 hours, all control animals died, whereas the phage-treated ones survived. And they were able to study this in very much detail, both microbiology perspective, looking at the tissue, statistical, seeing the difference between treated and untreated when comparing the different time points. But the point that I want to make here is that Laurent and his group specifically isolated phages for this bioluminescent PAC strain. And it was these phages which were perfectly able to protect the animal from the Pseudomonas infection. But when he used our phages, which were not selected or isolated specifically to this PAC strain, he saw that the specifically isolated phage were much more efficient than the general phages that were given to them and administered. These phages still were able to clear or help half of the animals survive, but clearly the distinction could be made. So also the in vivo efficacy is phage dependent and requires a tailored preparation most of the time. Yeah. The third aspect is processing and delivery. I'm going to be very brief about this. This is earlier work done by our group, in which phages were spray dried. Yeah. Spray drying allows you to make dry powder inhalers, And actually, the phages are engulfed in these small uh, globules of powder material, and depending on the size of those particles that are made, these particles are going to go in the upper airway or deeper or all the way down into the deepest reaches of the lung. And we can play around with the particle diameter to ensure an optimal deposition at the site where you want to release your phages. You have to realize this is really important because especially parents will know if you have to use vapor inhalers that can be very uncomfortable. So the capacity to do this as a dry powder inhaler has a huge benefits both towards efficacy and patient comfort. Now, the big question that we tried to answer was, can we introduce phages into those particles? Well, this depends again on the excipient solution, the drying parameters, the particle size distribution, and the powder stability. And all of these parameters were different for a specific phage. So all these phage processing towards dry powder inhalers are also strongly phage dependent. So that means that this preclinical assessment, when we're looking at preclinical efficacy, when we're looking at pharmacology, when we're looking at processing and formulation, is giving you an incredible funnel towards applicability of a certain phage for a certain patient under a certain condition. And that funnel makes it very difficult to expand phage libraries to be optimally used towards phages, towards patients. And that for me, this incredible specificity and the tailoring that requires, for me, precludes an off-the-shelf approach where you can simply generally buy something and that will work in all cases. Yeah, I think we're going to go to a more tailored, personalized approach where patients are tested and have optimal cocktails tailor-made for themselves. What the impact will be towards commercialization, towards approval, towards clinical trial design is going to be massive as well, and a key limiting factor. So in the third step, when we look at the regulatory framework, this is the typical, if you have a new antimicrobial, this is the standard approach where you have a product, you test it and can produce it, you do preclinical testing on animals, you go to phase 1, 2, 3 clinical trial, there is review and approval, marketing, phase 4 safety, and then you have your medicinal product made by a pharmaceutical company. And this is going to take several months to several years. This is incredibly expensive, costing hundreds of millions to even billions of euros. And you have a fixed product. Yeah. That can be useful for a COVID vaccine. We don't consider it useful, and at least the group of Pirne decided that, yeah, maybe we should have a more sur mesure, tailor-made approach, more flexible approach that can be achieved in a hospital or a phage therapy center that allows you to start from a bank of phages that has been quality controlled, small scale produced, establishing phage stocks that can be produced in a specific way in a lab, to select certain phages from this bank for an individual patient, which is the best one, then add it to a cream, hydrogel, or whatsoever, to then give to the patient. If the patient has resistance to phages, or if there is no phage available, new phages need to come in, either trained, banked, or the isolation of new phages that can go into this circuit to allow an individual phage treatment for a certain patient. Yeah. This has the advantage, if it's allowed to do it in such a way, that it could have a lower cost and a more rapid turnover. Now, this at the time when Pirne postulated this in 2011 was still not feasible. It was in this time that together we established a non-profit called phage. Phages for Human Applications Group in Europe, which was initiated very much in Belgium by the military hospital Neder-Overheembeek, Lab MCT, and Jean-Paul Pirne, Gilbert Verbeek, and Daniel de Vos. And that hospital specializes obviously also in burn wounds, has clean rooms that are used for tissue culturing, that are used as sutures for burn victims and so forth. So these clean rooms are the ideal facility to also create and generate, produce phage cocktails. And this is actually what was done for the first time in 2009 to create the first phage cocktails. And then after a lot of pushing to under the Declaration of Helsinki to treat an individual patient. The paperwork you see here was the paperwork needed to treat one patient. Taking several months to get approval, and this was only feasible in very specific cases where the MD made a judgment call that other therapeutic options were no longer on the table. And under this Declaration of Helsinki, a handful of patients have been treated in the past 10 years, let's say. So this was terribly complicated, and there was actually no established frame towards using phages. So we set up one ourselves together, a multidisciplinary approach focusing on directed microbiological research on the isolation and production of phages and assessing their validity, assessing the quality on a microbiological level, assessing the quality of the production, and then a final quality control towards safety, and to develop something that mimics a quality control that can satisfy legislator, can satisfy ethical committees. And this was actually the basis for the solution that is present now in Belgium, and it is the magistral phage. Yeah? And that relies on something that already exists for quite some time under European and Belgian law, and that is a magistral preparation. And the magistral preparation is actually any medicinal product prepared by a pharmacy in accordance to a MD prescribing magistral preparation for an individual patient. And inside a magistral preparation are active pharmaceutical ingredients. And in our case, there was a temporary provision that phages could be included in a magistral preparation. And for this, it had to conform to a monograph. And this monograph, and you can see it in the publication done by Pirne et al. In 2018, this monograph was only 3 pages. So that made it much easier, still under extreme circumstances, to find solutions for individual patients. Yeah? So how does it work in Belgium? So we have our phage bank of characterized phages that have been carefully selected, and gradually we're trying to increase the number of phages targeting bacterial species like Pseudomonas aeruginosa, Staph aureus, Acinetobacter, Klebsiella, and gradually expanding our library of characterized phages in this banking system that can be produced as individual active pharmaceutical ingredients according to this monograph, and this needs to be verified by an independent Belgium approved laboratory. Yeah. So the production is happening in Queen Astrid, yeah, based on the phages that have been characterized in part by us. And this then goes to an external QC, which is a government institute called Ciencias, that is providing to see that the production of these phages happened in a good way. This then moves to a hospital pharmacy to, based on a doctor's prescription, adding one or two selected phages that have been selected based on whether they can infect the individual strain of a certain patient, that is put together as a magistral preparation, possibly combined in a carrier like a hydrogel for individual administration. And this now works, and yeah, this is not going very much in international media, but it's allowing people in Queen Astrid and the various hospitals in Flanders who are engaging to treat about 20 to 30 patients a year with phages. So this Belgian model for the implementation of phage therapy is still very much on a case-by-case basis, but the magistral phage enables us to upscale these individual treatments for carefully selected patients. Yeah. But it still remains crucial to expand the data bank of therapeutic phages that are present. Also, and this is critically important, it provided the essential basis to convince surgeons and clinicians to consider phage therapy as a therapeutic option. And many phage biologists will know up to a few years it was very difficult to convince MDs of the validity of phages. But what's critically important is that a multidisciplinary approach is necessary to achieve this. We need the surgeons on board. We need the pharmacists on board. We need the production to be stable. We need an independent body to support this, and we need to feed the bank with new phages that are thoroughly characterized. And this combination, this framework is in my opinion, the only one that works. And obviously still, because of the limitation of phages that are there, these individual cases should be carefully and cautiously considered, and this is something that's coordinated or monitored at least by the Belgian FDA. So that brings us to a final step 4, and that's the clinical implementation and framework that we have set up, let's say, within KU Leuven and the affiliated university hospital of Leuven, where we had scientists focusing on IP and legislation. Obviously, we were contributing to the molecular microbiology and cocktail design, the banking and production, the pharmacology and processing with the pharmacists and the experts that can spray dry or that can create gels. And then the clinicians themselves, the surgeons, where within Leuven we have a number of surgeons that are willing or learning how to use phages for these individual cases. And this learning curve is very steep and very restricted. So a very small number of phages of patients can receive phage therapy, and the basic phages that are being used mostly is phage ISP targeting Staph aureus, which was isolated by the Eliava Institute in Georgia, which has the long history of using phage therapy. Some phages that were isolated in collaboration with my lab or with Georgia, and these phages were actually analyzed by us from a molecular microbiology point of view. This phage uses the LPS as the primary receptor to recognize the bacterial cell. This one PNM recognizes type 4 pili, allowing them to target a relatively small percentage of Pseudomonas isolates. For Staph aureus, the story is different. This ISP phage hits most of the Staph aureus phages that are present. And phages from this cocktail are being used on very special cases. For example, this case was a lady who, for over a decade, has had multiple surgeries and implants and associated with this Staph aureus infections. These cases get then referred to the university hospital, and then it's Professor Metzemakers who is an expert. And when you look at this mess and how this was solved with this clean hip replacement implant, phages were actually added during surgery on a sponge that biodegraded and was left inside. Also, these drains could be flushed with phages to allow further admission or admission of phages to that infection. And all of the cases that have been done so far actually have been successful. And with success, we mean that after many years of surgeries, the surgery at UZ Leuven with the presence of phages was the first surgery to clear the infection. And with clear the infection, I mean that after 1 year post-surgery, no resurgence of Staph aureus was observed. This has been done for published for 4 cases that have come to the university hospital. There are now several more cases that have been treated in this way, and that was published relatively recently by Jolien Onsea et al. In Viruses. So what's our role in this? Because to be honest, I'm not a clinician. I try to not look at the gruesome surgery pictures. What we are interested in is what's happening in these patients from a microbiological point of view. Yeah, can we learn something from the microbes that are present in this patient? Yeah, and this is the kind of data that we are gathering and seeing. So what you see here is 3 chromosomes and of sequenced bacteria, and the different strains are actually isolates from different points each time later and later on. So these bacterial genomes were fully sequenced, taken at various time points of a certain patient having a chronic infection. We are sequencing those and seeing which mutations, what is the genome dynamics that the bacteria are undergoing, what is the adaptive evolution as bacteria are co-evolving or are evolving within a patient. And this allowed us to see, on one hand, again, the importance of combining sequencing technologies, observing the mobility of prophages that are present, genomic islands that are activating or deactivating certain genes, mutations that accumulate, the loss of plasmids that are observed, and so forth. So this is microbiological monitoring, let's say, before treatment of phages. But now, with more phage treatments happening, we're also in a position when a phage treatment starts and other clinical interventions that are happening to track both bacteria that are present inside as well as phages. And is there an evolution towards insensitive mutants in vivo during phage therapy? How is the phage modifying itself in this patient environment? And in this specific patient, it was a series of multi-drug resistant Pseudomonas isolates from a 1.5-year-old patient suffering from septicemia post-liver transplant, who received intravenous treatment and actually obtained life-saving therapy results. By tracking this microbiology, by identifying the mutations that are emerging and comparing them to other in vitro assays, that will be of interest. Together with the lab of Druliskała in Poland, we're looking at the evolution of phage insensitive mutants in vitro, and also studying what's happening in there. So Druliskała group selected 5 clinical isolates, let them grow either planktonically or in a biofilm, then administered phages either individually or cocktails of 2 or even 3 phages, and then selected phage-resistant mutants. So bacterial cells that are no longer susceptible to one or combinations of 1 or 2 phages in vitro. So by taking these resistant mutants, phenotyping them and determining antibiotic resistance, phage typing for cross resistance, growth, virulence, and then taking the most efficient, the most important mutants based on phenotypical differences, over 200 of these resistant bacteria were fully sequenced and analyzed for the presence of SNPs and deletions. So I hope you see that this data set is very interesting and specific. Where are we observing mutations? We're observing mutations in genes important for phage infection, the LPS, the flagella, the type 4 pili as expected, but also the effect of prophages that is having an impact on pseudolysogeny. We're seeing large genomic deletions that are occurring, and I think this is valuable, by combining these data, by combining data that we're acquiring from mutations from patients, hopefully we can circle all the way back to step 1, to our machine learning models, and based on this, try to improve these models, not targeting simply the presence or absence of a certain genes or gene clusters, but zooming in to SNPs, to mutations that determine the success of phages infecting, yes or no. So overall, the number of successful phage therapy cases is growing increasingly. It provides the surgeons with hands-on experience, which enables them to properly design new protocols and implementations that are critically important for clinical trials, and actually in part probably explain the lack of success in clinical trials that have been done to date. Yeah. At the same time, these cases are a solid foundation towards the clinical trials, but they can help us understand outstanding questions regarding immunology, the pharmacodynamics of kinetics of phages, the inpatient resistance development are now elements that we can study and follow up on. So with this, I want to thank all of you for your attention. I want to specifically thank Cédric Lot. He's one of the PhD students working with us. Also Jolien Onsea, Jeroen Wagemans, but of course the entire team within Leuven. And also our other collaborators, Professor Huis van der Motor, and especially Professor Metzemakers, the surgeon, the team in the rest of Flanders, with first and foremost Dr. Spirne Merabishvili, De Vos, and Verbeke, Dr. Ciencias for the phage therapy cocktails production and the more clinical aspects. The work done on the in vitro mutagenesis was done with Professor Druliskała, where we have a very close collaboration linked to phage biology as well. And with that, I want to thank you for your attention. Also, very important, we're involved in one EU project, Antibiolab, to prevent implant-related infections with the AO Institute partners in Pisa and Berlin. As some of you may know, I'm very much engaged in the International Society for Virus of Microbes. We usually have biannual meetings that some of you may know, the Viruses of Microbes meetings. Usually they're every 2 years, but now due to Corona, these have been squished together. So in 2022, if you're interested in microbial virus research, you're welcome in Portugal. In 2023, we go to the Foundation of Phage Therapy Research in Georgia, Tbilisi. And in 2024, you can discover Australia and learn about phages and other things. So thank you for your attention, and I'm happy to take questions.
Rob, thank you for this marvelous talk. It was fascinating and inspiring. Can you hear us?
Yes, perfectly. Thank you.
No, thank you. It was very, very inspiring indeed. I was just thinking, based on limited knowledge, that it's almost a century since someone started to use some substances that accidentally prevented bacterial growth, and suddenly 100 years later, that is in overall a small period of time, we are at this stage that is, I don't know, for me that I'm not related to the topic, just mind-blowing.
Yeah, I should mention that this is the molecular discovery in the West. You have to realize that during World War II, and looking at the history, I think the former Soviet Union and Georgia was at that time using phages to give to the Soviet soldiers. And this phage research and phage therapy was still continued in Georgia, and a lot of the historical experience has been a valuable input towards translating this to Western applications, let's say. And I didn't formally acknowledge, but obviously the interaction with the Eliava Institutes is a cherished one, and one that we're working hard on to expand and continue.
Oh, great. Rob, so I'm gonna put some questions of different persons that have asked throughout the talk, and if anyone wants to ask anything in Spanish or English, I'll try to translate it.
I have some Spanish, but not
No, but
for science.
I'll make an effort to translate it decently. So Laura Bonofiglio from Buenos Aires says, do you think that without data mining model, it is possible to study interactions between phages and bacteria? And could this model would apply to license?
Okay, I think, of course, people have been using phages and phage cocktails without knowing the intricacies. It's still a black box. But if there are biological reasons that you can discover based on microbiological research, I think this is always going to be valuable. In terms of license, I think license are a lot more easy. License of gram-positive bacteria are enzymes that degrade the peptidoglycan layer. And for gram-positive bacteria, these are peptidoglycan layer on the surface of the bacteria are very specific, but not so specific as phages. So based on a certain subgroup or species, license are specific. In gram-negative pathogens, there is the protective outer membrane, whereas the peptidoglycan is conserved. So the license there are broad spectrum, and the specificity is given by their capacity to degrade or infiltrate through the outer membrane.
Thanks, Rob. And there's a related question from Juan Camilo Farfan. He says, what do you think about administering phage cocktails by giving one after another instead of the whole cocktail at once?
Yeah, that's a very interesting question. I know that the people in the Burn Wound Center in Brussels are big believers of this approach. I believe this is also based on work they have done with the UK partners to where they are tracking evolution and resistance development of individual phages and sequentially provided phages. Uh, it's an interesting idea. The question is, I don't know whether it will be better or not. It's an open question and one that deserves further study. But it makes sense to have complementary phages and give them sequentially or after each other. Thanks,
Okay,
Mark.
thanks, Rob. And there's another question later in your talk by Natalia. This kind of machine learning model are contemplating specific ecological parameters, just such as biofilm formation, and how viable seems
Yeah.
implementing bioengineering phages in human phage therapy.
Mhm. So the short answer is no. I think because the basic data sets that we are having are based on plate-based infection, yes or no. To answer the question on biofilm formation, we need to assess phage infectivity under those conditions or mimicking these conditions as closely as possible. Hopefully, the mutation analysis can help in this regard. Yeah. Bioengineered phages are an interesting concept. On one hand, it allows you to have a stronger patenting position. Because if you're engineering a phage, you're weaponizing it in a certain way to have a broader host range, to circumvent resistance, making it, medicinal engineered phage. Is certainly an option that's being pursued by several companies. It is possible. We have done some work in collaboration with the company on this, and the key element there will be the key limitation is the capacity to indeed introduce foreign genes and things like that. How that will translate towards regulation and perception, I think in these COVID times, if you're saying we're using a virus to treat a bacterial infection, sounds weird. But if you say we're using a genetically engineered virus to treat a bacterial infection, you will lose a lot of votes by people who are not really close in the field, let's say. But yeah, engineering is another way to go and another way to try.
Great, Rob. Juan Carlos has another question. Neutralization of phages by antibodies may be a drawback to phage therapy in certain cases.
Yeah.
There is a way to predict this outcome before administering a phage to a
Yeah.
patient.
So that's a very interesting question. So that's another benefit that we are now having from from patients being treated with phages, is that actually this neutral antibody neutralization can also be tested, and the very preliminary indications, as I gather from the people in the Burn Wound Center in Brussels, is that there is not much long-term neutralization of phages by antibodies. I think from a biological evolutionary perspective, this makes sense. Our body is accustomed in our gastrointestinal tracts to have phages present. as an integral part of the gut microbiome. Predicting this in advance, um, yeah, you can test serum in advance if a phage is being neutralized, yes or no. You can test this experimentally, but to predict this, I think is very difficult. Yeah.
Rob, there's two more questions. I got a couple more, but this one's from the public. Laura says, is there any way to track and prevent the possible negative effects of bacterial metabolites on the phages during in vivo trials?
Is there any way to track and prevent positive negative effects of bacterial metabolites on the phages? Yeah, I think, Laura, you are touching on a very interesting topic. I was going to ask you which bacterial metabolites, but I think bacteria are producing a lot of secondary or specialized metabolites to ward off other bacteria and which have all kinds of properties and form the foundation of antibiotics. Now, small molecules are being looked at towards phage infectivity. Whether it has an impact, I don't know. But
Great.
again, I mean, if you want to do research there, this is going to be a hot topic in the near future in the fundamental side, I think.
Okay, and there's one final question. I cannot. Does the application of a phage cocktail run the risk of affecting other bacteria in the human body the way antibiotics do? How would the use of phage cocktails differ in immunocompromised patients?
Yeah, more difficult questions. The first, maybe not. I think the incredible specificity of phages that I highlighted in the beginning, targeting only a subset of isolates within a bacterial species, offers the opportunity to save the natural microbiome or restore the natural microbiome that's present. What the role of phages is in like patients with Crohn's disease and which have a perturbed microbiome, whether the lack of diversity of phages that present is a cause of effect, that's not clear. But in any case, the high specificity of phages is going to result in much smaller reservoirs of resistance being developed. You have to realize an antibiotic that goes into the sewer, all bacterial species that are targeted by this antibiotic will work towards resistance development, start cross-sharing, and I think this high specificity of phages will not have that same level of resistance development on the long term. The use of phage cocktails in immunocompromised patients, I think, is going to be a critical one. From an ecological point of view, phages are not ultimate killers. They are not there to eradicate a bacterial species. Obviously, having a focused delivery of a high amount of phages can lead to a local eradication, but I'm afraid, or I think, and this is guesstimating, that there is some cleanup work for the immune system on residual bacteria that are brought down by phages, but not fully eradicated. So in immunocompromised patients, yeah, that issue might be a problem.
Rob, there's one more question, if you don't mind.
No problem.
Thanks. And the question is, how do you see the effect of phage on the immune system, and do you see the possibility to treat autoimmune and autoinflammatory diseases that have overreactive immune response with the phage therapy?
I will admit I'm not smart enough to understand the human immune system very well, and certainly autoinflammatory diseases. Um, what I can say here is that phages have been interacting with the human body, and when we think about for eons, for billions of for millions of years, uh, and yeah, that in one sense helps a bit. I know there are observations of both anti and pro-inflammatory responses by phages. Some phages can have a negative effect and deflect the immune system away from recognizing predatory bacteria that are present when we and but yeah, it's not a clear cut view that I think is present at this stage.
Rob, well, there's many thank you around, and really thank you for this really interesting talk. I have to say, from a personal perspective, I was not familiar with the topic, but it's just fascinating what can be done, and what you and your colleagues are doing sounds like at the very edge of science in this field. So thank you very much for sharing all of this information with us. Yeah, it was very nice to have you here in the seminar.
Yep, thank you.
Okay, so thanks to, lo digo en español, gracias a todos y a todas que nos acompañaron hoy en el seminario, y nos vemos en 8 días en otro tema de seminario sobre comportamiento animal. Nos vemos en 8 días y muchas gracias por estar con nosotros.